Buffalo grass is very widespread and commonly found in the Midwest region of the United States. We set out to determine why we do not see this organism in our home state of Virginia. Buffalo grass is native to North America and thrives in clay soils with a moderate amount of rainfall, making it an ideal choice for low-maintenance landscapes. It has a strong tolerance for alkaline soil conditions, and it is adaptable to a wide range of environmental settings.
Virginia, along with other southeastern states, see an increase in annual rainfall, making the soil more acidic. Knowing this, we are testing buffalo grass in a variety of pH conditions, predicting that we will see the least amount of growth in the acidic pH levels.
We altered our methods from Zhang, who examined soil pH effects on tobacco plants. We tested 4 different pH gradients, 5 through 8, with 7 as our control. We had 3 trials per pH level, each containing 30 buffalo grass seeds sprinkled on top. To achieve the desired pH levels, we used fast-acting lime to make the soil more alkaline and soil acidifier to make it more acidic. We ensured accurate pH levels with the pH meter instrument as well as pH strips. Once planted, we water the seeds twice a day with deionized water for the entirety of the project, as there was continual growth with that watering schedule.
We place the seeds in the greenhouse to provide optimal growth conditions, enclosing them in with clear shower curtain liners to prevent the sprinkler system from reaching them. To ensure stable pH levels, we measured the soil's pH using the pH instrument once per week. We collected data on the number of seeds germinated and average blade height over a 10-day period. Post-final data collection, we emptied the containers and placed the grass blades and their roots in the oven for 4 hours at 90 degrees Celsius to remove any excess water. We cut the blades at their root connection point and masked them.
Here's our data showing combined biomass across all trials. pH 7 had the largest biomass at 0.74 grams, followed by pH 8 at 0.53 grams, showing the most successful growth in the neutral to alkaline pH groups compared to the acidic conditions. Sample size represents the number of seeds that germinated per pH level.
The second dependent variable we looked at was total percent germination over time. With a population size of 90, pH 5 showed the highest rate of germination, but the lowest total germination percentage over time at 25. pH 7 showed the highest total germination percentage, around 45. pH 6 and 7 showed similar patterns over time, with the pH 7 numbers consistently being slightly higher than that of pH 6. pH 8 showed the lowest overall increase in germination rate over time. All pH levels showed an increase in germination over time, leaving us to believe that if kept in the same conditions, seeds would continue to germinate.
Finally, we examined blade height over time using the average of the tallest 5 blades. pH 7 showed strong linear growth and ended with our tallest average at 110 millimeters. pH 5, 6, and 8 were fairly stagnant from collection checks 1 to 2. Then pH 8 exponentially increased, having the second highest average at 72 millimeters. pH 5 and 6 both showed slow growth from time intervals 2 to 3, with 5 ending as our lowest average at 37 millimeters.
So our findings align well with our original hypothesis that acidic soil pH levels would result in reduced buffalo grass biomass, while neutral and alkaline pH soil levels would foster more robust growth. The biomass data showing pH 7 at the most favorable condition with 0.74 grams confirms that buffalo grass thrives in neutral to slightly alkaline soils, consistent with its natural habitat in the Midwest.
This supports Honecker's research on buffalo grass, which shows that the optimal pH is between 6.5 and 7.5. While the seeds can germinate in acidic conditions, pH 5 and 6, their growth is significantly reduced, suggesting they struggle to thrive in such environments.
A study on soil pH buffering capacity, PHBC, shows how critical of a determining factor it is in how soils resist changes in pH, which directly affects plant growth and ecosystem stability.
So our overall findings show buffalo grass thriving at neutral to slightly alkaline conditions, which agree with the idea that soils with higher PHBC support stable growth. This is particularly relevant when considering southeastern acidic soils and the higher rainfall that they receive. As the study indicates, such conditions reduce the soil's ability to buffer against acidity, creating a less favorable environment for buffalo grass.